Welcome to my YouTube channel. I am excited for you to be here today. I am Sharoon Daniel and we will continue our walk through our past papers, and today we will discuss further questions of Foundational Paper 2 GCSE Biology AQA exam. Let's begin, and if you like my content, please like, share and subscribe for more videos and resources. Now let's begin with first question, which is question 5, and it is all about decomposition and decay. Here examiner is interested in checking your knowledge rather than just the memory recall. So let's start with first part of question 5, which is 5.1. Let's read this question together, and it asks to name one other organism which decompose and decay, and the answer is fungi. Along with bacteria, these are the main decomposers. And now let's take a look at next picture. Figure 8 apparatus is shown, and the question is 5.2. What is the dependent variable? And the answer to this question is the pH of the milk. I will give you an exam tip that the dependent variable is the one you data collect measure. The independent variable is the one you change. Temperature. Similarly, next part of the question 5.3 is about control the temperature to specific temperature. And answer to this scientific question is put the beaker in a water bath. Exam tip here is that the water baths are the standard way to keep a solution at a precise, constant temperature in biology. Next part of question 5.4 asks you what type of acid is produced by digestion of fats in milk. And the answer is fatty acid. Exam tip. Fats, lipids, are broken down into fatty acids and glycerol. The production of these acids is why the pH drops as milk spoils. Figure 9 shows a line graph, and the part of question 5.5 asking you scientific reason why at 20 degrees pH does not fell under 4.5. Answer. All the fat had been digested. Exam tip. When a graph levels off, goes flat, it usually means the reactant has been used up or the reaction has finished. Now moving on to next part 0.56, which asks you one reason why rate of digestion is faster at 30 degree rather than 5 degree, and the answer 5.6. Reason for faster digestion at 30 degrees Celsius. The primary reason is that enzymes and substrate molecules have more kinetic energy at higher temperatures, up to the optimum. The reason, as temperature increases, molecules move faster. This leads to more frequent successful collisions between the lipase enzymes and the fat molecules, resulting in more enzyme substrate complexes being formed per second. Hence at 30 degrees is the optimum temperature for enzymes to work at maximum level. Next question required you to read graph in figure 7 at 30 degrees where the pH of the milk changes from day 2 to 3 according to the question part 5.7 to find out rate calculation, and the answer is the pH at day 2 at 30 degree was 6.0. The pH at day 3 was 4.7. Now let's calculate, and the fall in pH 6.0 to 4.7 equals 1.3 pH units day, in the final answer. Final question, which is 5.8, is about comparing rates, as this question asks for a ratio. You are comparing your answer from 5.7 to the given rate of 0.1 pH units day. The formula for this calculation is rate at 30 degree divided by rate at 5 degree. Example, if your answer for part question 5.7 was 0.4, the calculation would be 0.4 divided by 0.1 equals 4. You would write 4 times faster. Let's begin our next slide showing